Good morning, good afternoon, and good evening, everyone. And a very warm welcome to the Scientist.com and Stem Cell Technologies Translational T-Cell Talks. I'm Liam Sanyo from Scientist.com. It's been my pleasure to work with the Stem Cell team on webinars on a whole range of topics, but today's half-day event is all about T-cells, and especially in the CAR T space. And with that, I'm very pleased to welcome the first speaker, Dr. Uslu, who goes by Oogie. So Oogie, thanks so much for joining us today. And the floor is yours whenever you're ready.
Thank you, Liam, so much for the kind introduction. And thanks a lot to Stem Cell and the scientists for inviting me for this event. So there are amazing talks today, and I'm honored to be the first one speaking today. So I'm going to talk on CAR T cells for the use in solid tumors. Just very briefly on my background, I'm a trained dermatologist in Germany. And besides my clinical training, I worked translationally on dendritic cell cancer vaccines and CAR T cells in melanoma and other solid tumors. And currently, I'm a postdoc in Carl June's laboratory at Penn, where I continue my translational work on CAR T cells, especially CAR T cells in solid tumors. I think all of you have heard of CAR T cell therapy. Just very briefly, in CAR T cell therapy, you take patients' T cells. In the laboratory, these T cells are engineered to express an additional receptor. So this receptor is called a CAR. It stands for chimeric antigen receptor. These CAR T cells are then being expanded and then reinfused into the patients. And since we have equipped now these T cells with this CAR, these CAR T cells can recognize the tumor and can eliminate very effectively tumor cells within the patient's body. And a chimeric antigen receptor is a synthetic receptor, which means in the nature or in the patient's body, you don't have CARs. They have to be synthetically built in the laboratory. They consist of two main parts. The intracellular part of the CAR is derived from a T cell receptor. The extracellular part of the CAR is derived from a monoclonal antibody. And therefore, CARs can recognize cell surface antigens, which are presented on the surface of the tumor cells without MHC, which represents an advantage over TCR T cells, for example. And those CAR T cells are being very effectively used for certain hematological malignancies. My mentor, Carl June, he developed the first CAR T cell product, which was approved, Chimera, back in 2017, and there are already 10-year data of the treated patients. So this data was published 2 years ago in Nature. And what is pretty amazing is that in all of those patients which were treated, after 10 years, you can still detect the CAR T cells in the blood of those patients, which is shown here in the dot plot. So in this patient, after 10 years, there's still 1.3% of the T cells, which are CAR T cells in the patient's blood. So that's why CAR T cell therapy is called a living drug. So basically you give one intravenous dose of the CAR T cells, and this one dose can provide a long-term protection from the cancer, at least in some patients, not in all, but in some patients. And one of those patients is Emily Whitehead. She's shown here on the right side. So she was the first child to receive this treatment. She didn't have any treatment options anymore at the age of 7, received, back then, it was a very highly experimental treatment. So she received the CAR T cells, and since then she's cancer-free, and now she's an undergrad at Penn. She's basically going to college now at the institution where she received the CAR T cells 10 years ago. And also beyond hematological malignancies, recent studies have shown that CAR T cells can also be used very effectively in autoimmune disorders, in systemic lupus, for example, or systemic sclerosis. So this is a recent publication from my home institution in Germany, actually. So I received my medical training and my scientific training in Erlangen, which is in the southwest of Germany. And this publication is from the group in Germany who first treated autoimmune diseases with CAR T cells. And it's a case series of 15 patients with very severe autoimmune diseases, including systemic lupus. And shown here on the right side is patient number 1. She's a young lady who had basically an end-stage lupus. She was taking about 50 pills a day, had kidney involvement, lung involvement, multiple organ involvement because of her lupus. And after the one infusion of the CAR T cells, she has been free of disease now for 3 years. And behind her are the treating physicians, Andreas Mackensen and Georg Schett, who are leading this clinical trial. So besides hematological malignancies and most recently also in autoimmune diseases, there is a big effort to also move CAR T cells into solid tumors. But in solid tumors, it hasn't worked that well yet, at least. So there are some successes, but we need to further work on that. And challenges of CAR T cell therapy in solid tumors are different. For example, one of the problems is the CAR T cell migration or the trafficking. So that means that the CAR T cells, they need to leave the blood vessels and find their way from the blood vessels to the solid tumors, whereas in hematological malignancies, the cancer cells are in the blood vessels. So that's different. Once the CAR T cells left the blood vessels and found their way to the solid tumors, the next hurdle is the immunosuppressive tumor microenvironment. So within the tumors, there are immunosuppressive tumor immune cells, for example, Tregs or MDSCs, and also immunosuppressive cytokines like TGF-beta. And this immunosuppressive tumor microenvironment can make the CAR T cells get exhausted so that they don't function well and cannot fight the tumor properly. And once they start fighting the tumors, another problem is immune evasion mechanisms. That means that the tumor cells, they can lose the antigen, which is recognized by the CAR. And if that happens, the CAR T cells will not be able to fight the tumor cells anymore, which will result in tumor progression. And my scientific work focuses on mechanisms to potentially overcome these challenges. And today I'm going to present you for each of these problems one potential strategy. As a strategy to overcome the first problem, the CAR T cell migration, you could apply the CAR T cells locally at the tumor site rather than intravenously, because if you don't give the CAR Ts IV, but if you apply them locally, directly where the tumor is, the CAR T cells don't need to find their way because you already applied them there. And one of the projects I'm working on in Carl June's lab is the intraoperative local use of CAR T cells. So the surgical excision is basically a very important part for treatment of most solid tumors. And the goal for the surgical excision in solid tumors is to basically cut out the whole tumors so that there are no residual tumor cells in the patient's body. But this goal is sometimes not achieved, and the results are positive surgical margins, which is shown here on the right side of the graph. That occurs basically when the cancer cells are still present at the edge of the resection specimen. And reasons for positive surgical margins are that you sometimes can only see the positive margins under the microscope, but not during surgery, so it was not palpable or visible at the time of surgery. And another problem is, or a reason for positive margins, and that's a major problem for locally progressed breast cancer, for glioblastoma, for example, or for pancreatic cancer, that there are very crucial structure and the anatomic location which does not allow you to get negative margins, basically. So you will always, in pancreatic cancer, you'll always have positive surgical margins. And our idea was to apply CAR T cells locally in an intraoperative setting as an approach to basically cut out as much as possible and then apply the CAR T cells so that the CAR T cells get rid of the residual tumor cells. And that's what I tested in mice. So we first established a partial surgical resection model. So we injected the tumor cells into the right flank of NSG mice. NSG mice are immunocompromised mice. They don't have adaptive immunity, so that you can inject human tumor cells and human T cells. And we first injected these human tumor cells in the right flank of those mice and then surgically removed about 75% of the tumor to mimic incomplete surgical resection. So we mimic the R2 resection. So basically, when you leave a bigger part in the body, it's called R2 resection. And then the next step, we apply the CAR T cells intraoperatively so that the CAR T cells will get rid of the rest of the residual 25% of the tumor. And before we apply the CAR T cells intraoperatively, we first mix them in a fibrin glue-based carrier so that the CAR T cells basically will get stuck. I don't want to say stuck, but so that they persist longer intraoperatively on the wound, so that we could apply the CAR T cells into the resection cavity on the wound so that the CAR T cells basically persisted longer. So that was the idea of first mixing the CAR T cells in a fibrin glue-based carrier. And shown here is the overall survival of the treated mice. So we tested this approach in 2 xenograft animal models. So MDA MB231 is a triple negative breast cancer cell line. So one of the models was a human triple negative breast cancer model. The second model was a pancreatic cancer model using the human pancreatic cancer cells ASPC1. And in red are shown the mice, the survival of the mice which received the surgery following by the CAR T cells in the fibrin glue-based carrier. And you can see here in both models that these mice significantly lived longer when compared to the green line. These mice also received surgery and the CAR T cells, but without the fibrin glue. They just received the CAR T cells basically in a liquidy media solution. So that shows us that the fibrin glue really makes the CAR T cells stay longer on the surgery site. And the next thing we wanted to test is, since we apply the CAR T cells intraoperatively, we don't want the CAR T cells to interfere with wound healing. So basically, you have an open wound, you apply the CAR T cells, and then you close the wound, and then you also want the wound to heal properly. To test that, we repeated the animal experiments, and we used a syngeneic mouse model using black 6 mice. And syngeneic mice, they do have their own immune system. They are immunocompetent mice, and that will basically allow us to really see if there are wound healing complications. And shown here is one mouse, one representative mouse, which only received the incomplete surgery, the removal of 75% of the tumor at day 1 after surgery. You can see here the surgical clips. So we used clips to close the wound, but you cannot see, we didn't see any redness, swelling, wound healing complications, or any signs of wound healing complications. At day 6, when we removed the surgical clips, it was a similar picture, no signs of wound healing complications. Also, a few days later, there was no signs of any redness or swelling. You can see here that the tumor regrows, so that was because we only removed 75% of the tumor. That means that the 25%, which is still in that mouse, will of course relapse and regrow. When we show at a representative mouse which received basically surgery followed by the CAR T cells in the fibrin glue-based carrier, or we call it fibrin gel, you can see a similar picture. 1 day after surgery, there are no wound healing complications, no redness, no swelling, no pus formation, and the same at day 6 and also at day 10. So here you don't see any residual tumor cells because the CAR T cells, which we applied intraoperatively, they were able to get rid of the residual tumor cells. And in summary, we could basically show that CAR T cells can be safely used as an adjuvant therapy following incomplete surgical resection. So we showed the feasibility in 2 xenograft animal models, which I showed you in the previous slide. We could show in syngeneic mice that there was no wound healing complications. And we also showed that the local application of the CAR T cells resulted in reduced toxicity when compared to IV-infused CAR T cells. So we tested this in a transgenic cytotoxicity mouse model, which I couldn't show today because of time reasons. But the paper is published. We published it last year at Science Advances. Please feel free to have a look at the paper and reach out to me if you have any questions. And based on this data, there's also a clinical trial right now, which is already recruiting patients. So hopefully we can also help patients with this approach. So to come back to the overview slide. So the second problem of immunosuppressive tumor microenvironment. And here is a very attractive approach that you combine CAR T cells with other treatment options, basically to first apply the other treatment to change the tumor microenvironment and then infuse the CAR T cells in order to basically make the CAR T cells traffic better and also function better within the tumor microenvironment. And this is another project I'm working on in Carl June's lab. So what we did is to combine CAR T cells with a new STING agonist. And this is a collaboration project between our lab and James Chen's lab. James Chen is very big in the field of STING agonists, and we are basically combining his STING agonist with our CAR T cell products to see if this will basically help CAR T cells to function better. What is the STING pathway? So the STING pathway is a pro-inflammatory pathway, and there are different molecules involved in this pathway. So the cGAS first generates the molecule cGAM, and cGAM then binds to the protein STING, which stands for stimulator of interferon genes. And this STING then triggers the expression of type 1 interferons and inflammatory genes. So like I already said, it's basically a pro-inflammatory pathway. And by activating this inflammatory pathway, you can activate the innate immune defense and also induce the adaptive immune response. And for cancer treatment, you actually want to have an inflammation of the tumor. So that's why STING agonists, basically drugs, in order to activate the pro-inflammatory pathway, have been tested in tumor models preclinically and also in patients already. So there are like two products which are where they were pretty far in the clinical development, the MXAA and CGAMP. And we could see, or the colleagues could see, that basically the STING agonist, by inducing the inflammation, it is possible to change the tumor microenvironment. So you could turn basically immunologically cold tumors. So that's what immunologists like to call tumors which are lacking T cells or other pro-inflammatory immune cells, and you can turn these cold tumors into hot tumors, and hot tumors are more likely to work with immune checkpoint blockade, for example. But unfortunately, like by using these first generation STING agonists, you could not really see an anti-tumor effect. So that's why further improvement was needed for the development or to move STING agonists into solid tumors. And that's what James Chen did. So he developed a new STING agonist, which is called IMSA-101. And that new STING agonist, the second generation, basically, is a synthetic small molecule analog of CGAMP. And it shows superior serum stability in patients and also superior anti-tumor efficacy compared to conventional CGAMP. And this we tested in an animal model. So we injected into black 6 mice B16 tumor cells, which is a mouse melanoma cell line, into the right flank of those mice, and then treated those mice either with the new STING agonist, the IMSA-101, or with the conventional first generation like CGAMP product, basically. And we could see at both doses, so we injected either 3 micrograms or 10 micrograms, you can see at both doses that the mice receiving the new STING agonist showed significantly improved overall survival when compared to mice receiving the conventional CGAMP. So this basically supports that the new STING agonist is more potent, and based on this data, there is already clinical trials running to test IT STING agonists with different combination treatments, with radiation therapy, with immune checkpoint blockades, for example. And our aim is basically to also combine this new STING agonist with our CAR T cell products to hopefully make the CAR T cells work better in a solid tumor setting. So that's basically what I tested in syngeneic mice. So we used here black 6 mice, which have still their immune competence, so they have their own immune system, so that we could really monitor and test how the STING agonist is changing the tumor microenvironment. And we first injected basically the STING agonist intratumorally, so that we could change the tumor microenvironment. And the next step, we infused the CAR T cells intravenously, and we hoped that basically the CAR T cells will traffic better and function better since we first treated with the STING agonist. And when we show here at the overall survival, we tested this in 2 syngeneic mouse models. So basically, one is a mouse pancreatic cancer cell line, the PDA7940B. So this is a very cold tumor model. The CAR T cells alone, they basically don't really traffic into this tumor model because it's so aggressive and so cold. It is derived from KPC mice. And our second tumor model is a B16 model. So this is the mouse melanoma cell line, which is a warmer tumor model and a more immunogenic tumor model. And our second tumor model was basically to confirm the findings from the first tumor model, to just basically show that this approach not only works for one tumor entity, but also for different tumor entities. And in green is the overall survival shown of mice receiving the STING agonist, followed by the CAR T cells. And in both animal models, you can see here very nicely that the combination treatment, like mice receiving the combination treatment, showed significantly improved overall survival when compared to mice receiving the STING agonist alone, which is shown here in blue, or the CAR T cells alone, which is shown in red. So basically, we could show here in 2 animal models that the combination treatment really works better than the monotherapies. We then wanted to look a little bit deeper to the tumor growth. So shown here is basically the same mice, but we plotted here a tumor volume over time to see like how the tumor is growing and basically if there are complete remissions. On top is the tumor growth shown of the PDA tumor model. At the bottom of the B16 tumor model on the very right, we can see mice receiving the combination treatment. And here you can see that in the PDA model, there were 3 out of 10 mice which showed a complete remission. So the tumor disappeared following the combination treatment. In the warmer tumor model, the B16, which is more immunogenic, we could achieve a complete remission of 100%, basically. Like all of the treated mice, 10 out of 10 showed a complete remission. The tumors disappeared. When we look at the body weight, so the mouse body weight over time is always very important to measure, because that's like the only way to really tell if the mice, or to objectivize if the mice are doing well, because of course you cannot ask them how they are doing. You can of course observe them if they are moving slower, if they have like any clinical signs of distress, but like measuring the body weight will really tell you if they are doing good or not. If they don't do well, they will lose weight, and then we need to also sec them sometimes if they lose too much weight. On the very right side is the tumor weight, basically, of the mice receiving the combination treatment. On the top, again, the PDA, at the bottom, the B16, and we could not see any significant weight loss in all of the mice which received combination treatment, and also clinically they were moving around and running around. So this is basically very important to us, because we can see that the combination treatment has a very strong anti-tumor effect with complete remission in some mice in the PDA model, or in all mice in the B16, but none of the mice showed body weight loss, so all of them tolerated the treatment very well. So based on these data, we can conclude that combining CAR T cells with this new STING agonist improves the tumor control and overall survival in 2 syngeneic mouse models. And next, we wanted to understand mechanistically better, like how is the STING agonist basically changing the tumor microenvironment, leading to an improved CAR T cell functionality. In order to test that, we repeated the animal experiments. We took out the tumors 1 week after treatment, prepared the tumor single cell suspension, and run a flow cytometry. So in flow cytometry, we looked for the CAR T cells. So shown here is the count of the intratumoral CAR T cells. On the left, the PDA tumor model, on the right, the B16 model. And in both animal models, we can see that there are more intratumoral CAR T cells in the combination treatment group, which is shown here in green, when compared to the CAR T cell alone group, which is shown in red. We wanted to confirm our findings from the flow cytometry with a second method. So we performed the RNA in situ hybridization, the RNA-scope, in order to detect CAR transcripts in formalin-fixed paraffin-embedded tumor tissues. And you can see here in this representative image in pink is basically the CAR transcripts, where you can see that there are CAR T cells basically in the tumor tissues. And our pathologist, he quantified the data, and you can also see in the RNA-scope, which is consistent with the flow cytometry data, that the tumor tissues which from the combination treatment group, we could detect basically significantly higher levels of CAR transcripts when compared to the tumor sections which were treated with the CAR T cells alone. So based on the flow cytometry data and also the RNA-scope data, we can basically conclude that injection of the STING agonist leads to an increased trafficking of CAR T cells into the tumors. We also looked in the tumors for other immune cells. So this is the flow cytometry data shown for different immune cells in the tumors. So endogenous T cells, B cells, NK cells, macrophages, and MDSCs. On top, the PDA model, at the bottom, the B16 model. And you can see here that there are always more immune cells in the combination treatment group in green when compared to the CAR T alone group, but not between the combination treatment group and the STING alone group. So basically the green dots and the blue dots, they're always like higher levels of these two cohorts, pretty much throughout all of the immune cells. So this basically shows us that it's the effect of the STING agonist. So when you inject the STING agonist, either alone or in combination with CAR T cells, it seems to really increase the trafficking not only of the infused CAR T cells, but also of other immune cells in general. And in order to confirm our flow cytometry data, we used a second method to confirm this data. So we submitted our formalin-fixed paraffin-embedded tumor tissues to the pathologist, and our pathologist performed IHC stainings to look for different immune cells. So he looked for CD3 positive cells, or T cells, NK positive cells, F480 for macrophages, and Ly6G for neutrophils. And we could basically confirm our flow cytometry data with the IHC data. The pathologist also quantified it. And here it's also, you can always see more immune cells in the combination treatment group when compared to the CAR T alone group, but no differences basically between the combination treatment group and the STING alone group. So for NK cells, Ly6G neutrophils, macrophages, and also a trend in the T cells. When we look at representative images, Plotted here on top is the Ly6G. It's neutrophils shown, at the bottom the macrophages, and positive cells is basically the brown color. And when you look at untreated tumors or tumors treated with the CAR T cells alone, you can see positive cells basically at the tumor edges, like surrounding the tumor, but there are no positive cells inside of the tumors. But when you look at the STING-treated tumors, either STING alone or STING with CAR T cells, you can see that there is really a massive infiltration. You can see a lot of brown color inside of the tumors, which basically confirms the flow data and also the IHC data, which is quantified on this side. So based on this data, we can basically conclude that the STING agonist remodels the tumor microenvironment, leading to an increased infiltration of CAR T cells and other immune cells into the tumors. And next, we wanted to have a closer look to the intratumoral CAR T cells. We wanted to understand basically how the CAR T cells are changing when you additionally inject the tumors with the STING agonist. So we repeated the animal experiments. A week after treatment, we removed the tumors again and prepared the tumor single cell suspension and ran a flow. And we just gated on the intratumoral CAR T cells. We first wanted to find out the CD4 and CD8 ratio of the CAR T cell counts inside of the tumors. And on the top is basically the CD4 positive cell count shown. At the bottom, the CD8 positive cell count of the intratumoral CAR T cells. On the left, the PDA model, on the right, the B16 model. And very interestingly, when you look at the tumors of the CAR T alone group, you can detect significantly higher levels of CD4 T cells, which are the T helper cells. But when you inject the STING agonist, you see significantly higher levels of CD8 T cells in the tumors of the combination treatment group. So basically, what the STING agonist is probably doing is to basically really attract or help CD8 positive T cells rather than CD4 to traffic into the tumors, which are basically the cytotoxic T cells. And that's probably also like one reason why we see a better tumor control, because there is basically an increased infiltration of the CD8 CAR T cells into the tumors. And we also calculated the CD4-CD8 ratio here. So this is basically the same data which I described on this side. You can see basically it confirms that basically there is significantly higher levels of CD4 T cells in the CAR T alone groups when compared to the combination treatment groups, which had significantly higher levels of CD8 positive CAR T cells in the tumors. We also looked for the phenotype of the intratumoral CAR T cells. And we could see in both animal models, the PDA on the left and on the right, the B16, that there were significantly higher levels of effector T cells in the combination treatment group. And effector T cells are known to have basically a higher cytotoxic function. So this correlates with our observation that there are significantly higher levels of CD8 T cells, basically. So that was pretty interesting to see that basically the STING agonist primarily seems to attract CD8 T cells. We then wanted to basically further understand the role, or basically how the STING agonist is changing CAR T cells in the tumor. So we isolated the intratumoral CAR T cells and compared the gene signatures of the intratumoral CAR T cells of the combination treatment group compared to the intratumoral CAR T cells of the CAR T alone group. We used the nanostring ENCOUNTER platform, and the ENCOUNTER allows us basically to analyze about 800 genes, and you can choose different panels. And we chose a panel which had mostly T cell activation associated genes inside of the panel, because we were interested to see if the STING agonist really activates the intratumoral CAR T cells. And when you look here in the volcano plot, you can see that there are more than 200 genes which were significantly upregulated in the combination treatment group when compared to the CAR T alone group. And when you look in particular for activation associated genes, so this is an activation signature which Marcella Maus described in one of her prior publications in 2020. And we also had like most of those genes which Marcella Maus described also in our ENCOUNTER panel. And we could see that the intratumoral CAR T cells of the combination treatment group had significantly higher levels or upregulation of the interferon alpha gene, PD-1 gene, IL-2 gene, TIC, TIGIT, granzyme B, and other pro-inflammatory and activation associated genes. So this basically shows us that adding the STING agonist to the treatment seems to basically not only increase the infiltration of CD8 positive T cells into the tumors, but also seems to activate the intratumoral CAR T cells. We then also looked for pathway analysis. So we looked for different pathways of the intratumoral CAR T cells, and we could see that primarily activation-associated pathways were significantly upregulated in the combination treatment group, whereas primarily anti-inflammatory pathways were significantly down, like, for example, TGF-beta. And our attention got the IL-18. So basically the most significantly upregulated pathway in the combination treatment group in our analysis was the IL-18. And IL-18 is known to significantly improve CAR T cell function and CAR T cell expansion. So that's basically a cytokine which we analyzed in the laboratory for many years already in Carl June's lab. And we know that IL-18 is really improving CAR T cell function. There's also a clinical trial right now running with CAR T cells secreting IL-18, and those clinical trial results are really amazing, which I cannot share today because it's preliminary data. But we know that IL-18 really improves CAR T cell function, and based on this transcriptomics data, we hypothesized that maybe the STING agonist induces IL-18 secretion and thereby enhances CAR T cell function. And in order to test this hypothesis, or to basically show that the STING agonist is indeed inducing IL-18 secretion, we checked for cytokines in the tumors. So we performed a Luminex assay where we could analyze 17 cytokines of the tumors. And it's a logarithmic scale. That's why the differences don't seem to be that big, but it is statistically significant. And when we look at IL-18, we can here indeed see that the tumors of the combination treatment group showed we could detect significantly higher levels of IL-18 when compared to the CAR T alone group, but not between the combination treatment group and the STING alone group. So this is basically consistent with the immune cell data, which I showed earlier in IHC or flow cytometry. So this also seems to be an effect of the STING agonist, basically either alone or in combination with CAR T cells. It seems to induce IL-18 secretion. But it's not only IL-18. There was really differences seen in different, primarily pro-inflammatory cytokines, for example, IL-1 beta or interferon gamma and TNF, where we could always see in the two cohorts, either the STING with CAR T cells or STING alone, there were like higher levels throughout all of those cytokines, basically. But where is the IL-18 coming from? So we looked into the literature, and we found that basically the primary source of IL-18 secretion are the macrophages. So we could already see in the IHC data and the flow cytometry that the STING agonist seems to induce trafficking of macrophages into the tumors. So we also thought probably it does not only induce trafficking of the macrophages in the tumors, it also activates the macrophages so that the macrophages secrete more IL-18. In order to test this, we performed the RNA in situ hybridization, where we basically looked for IL-18 transcripts and also for macrophage transcripts. So ADGRE1 is the transcript of F480. It's basically for macrophages. And you can see here, our pathologist quantified it again. You can see, which confirms our cytokine data, which I showed in the previous slide, that in both animal models, in the PDA as well as in the B16 tumors, we could detect significantly higher levels of IL-18 transcripts in STING agonist treated cohorts when compared to control tumors which did not receive the STING agonist. And when we quantified the macrophage transcripts, we could also see here, which supports or which confirms our flow data and the IHC data, that there were higher transcripts for macrophages in the STING-treated cohorts in both animal models when compared to control tumors. So this basically confirms that there are more IL-18 in the tumors of the STING-treated tumors and also more macrophages. And our pathologist, he designed an algorithm to basically also see if the count of IL-18 secreting macrophages is higher in the STING-treated cohort. So that's what we plotted here. So this is basically the IL-18 transcripts in the macrophages. And we can see in both animal models, in the PDA as well as B16, that the STING agonist seems not to only increase trafficking of the macrophages into the tumors, it also seems to increase the level of IL-18 secreting macrophages in both tumor models. So based on this data, we could basically conclude that the STING agonist induces a secretion of IL-18, which we could show basically in RNA scope, in Luminex cytokine data, as well as the transcriptomics data of the pathway analysis. And in order to test our hypothesis if the STING agonist-mediated IL-18 secretion also induces CAR T cell function or improves CAR T cell function, we repeated the animal experiment where we compared wild-type CAR T cells with IL-18 receptor knockout CAR T cells. And these IL-18 receptor negative CAR T cells, we were hoping that they will perform worse because they don't get the help over IL-18 since they are lacking the receptor. And that's also what we saw. Shown here is the overall survival. So when we look at the mice receiving the STING agonist with the wild-type CAR T cells, they lived significantly longer when compared to mice receiving the STING agonist followed by the knockout CAR T cells. But when we look deeper into this data, we can see that there's also a significant difference between the STING agonist plus knockout CAR T cells versus STING agonist alone. So this basically tells us that IL-18 is one of the mechanisms enhancing CAR T cell function, but not the only mechanism. And you can see the difference is also nicer when you look at the tumor volume over time. When you look at the tumor growth of mice who received the STING agonist alone, we saw complete remission in 3 out of 12 mice. So the tumors disappeared in 3 mice. When we look at mice who were treated with STING agonist followed by the receptor negative CAR T cells, there were 7 out of 12 complete remissions. And mice receiving the STING agonist with wild-type CAR T cells, we saw complete remission in 100% of the mice. So that confirms basically that the knockout CAR T cells really performed worse when compared to the wild-type CAR T cells. And here again, if you look at the combination treatment group, we could not see any body weight change. So basically the treatment works pretty nicely and does not cause any side effects in those mice. So based on this experiment, we could basically conclude that STING agonist-mediated IL-18 induction enhances CAR T cell function. And that brings me to the end of my talk, or almost the end, actually. So the combination treatment of the STING agonist with CAR T cells improved overall survival in 2 animal models. And what we also did is we re-challenged those mice who showed a complete remission with the tumor cells to look for memory formation. And we also tested abscopal effects in a dual flank model. So I couldn't show the data because of time reasons, but we could basically see that the combination treatment not only improves overall survival, but also induces memory cell formation and abscopal effects. So please feel free to look into the publication. We recently published this in Nature Communications, where you can also see the data of the abscopal effect, for example. What I showed today is that the STING agonist increases trafficking of CAR T cells and other immune cells. So we could really detect significantly higher levels of different immune cells when we treated the tumors with the STING agonist. We could also see that activation-associated genes were upregulated in the intratumoral CAR T cells of the combination treatment group. And we could see very nicely that the STING agonist induced IL-18 secretion. So we could see this in the cytokine data of the tumors. We also checked the cytokines in the serum and the RNA scope data. And in our transcriptomics data, we saw that the treatment with STING agonist resulted in upregulation of the IL-18 pathway of the intratumoral CAR T cells of the combination treatment group. And this STING agonist-mediated IL-18 induction enhanced CAR T cell function in an animal model in vivo. So coming back to this overview slide, so the last challenge which I presented was mechanisms of immune evasion. So because of time reasons, I cannot go further into this, but I worked on this for several years in Germany. So this is like basically my data from a few years ago. So please feel free to have a look at these publications where we basically tried to engineer, for example, the CAR T cells not only with 1 receptor, but with 2 receptors so that the tumor has to lose both antigens and not only one, basically to make them to make it more difficult for the tumor cells to evade, and other strategies which we tested basically. Yeah, that's it. So I want to thank, of course, my mentor Carl June. He's an amazing mentor. I'm really happy to be part of his team and his laboratory. I want to thank Gina Young. She's a scientific director of the lab who's always supportive and helps wherever she can. And I want to thank the whole June lab, of course, especially the ones listed here who actively help for the projects, and also our collaboration partners who are listed here. Yeah, and please reach out to me on X, LinkedIn, or via email if you want to collaborate. So I'm happy to basically collaborate or to discuss science with you guys. So yeah, that's it from my side. actually. So thanks again for your attention and for your, for the invite.
Excellent. Well, thank you so much, Ugi, for a fantastic presentation. And with that, we'll move right into the Q&A. So, Ugi, ready to answer a few questions? Oh, I think you might be muted.
Let me see. Okay.
Yeah,
okay.
There you go.
Now I'm muted. Sorry. So, but I already saw like a few questions. So there are actually a lot of questions in the Q&A box, which is great. So, like I'll just start to answer them maybe. So basically one question is from Nesta Sang, do you apply the STING agonist and CAR T cells on the same day? So we did 2 STING agonist injections. The first injections was before the CAR T cell infusion. So in the morning, the STING agonist, in the afternoon, the CAR T cells, and the second STING agonist injection was at day 3, basically 3 days later. Then let's see, there is how much cardiotoxicity or nephrotoxicity does the association of this type of treatment with CAR T cells or IMSA 101 has? So I did not test this in animals, but our CAR T cell products, the mesothelin specific CAR T cells, which are used in these models, as well as the STING agonist alone, are already being tested in clinical trials. So for the STING agonist, we know that the IT injection of the STING agonist in the clinical trials, in the phase 1 trial, was safe. So there were no severe side effects. I don't think that they saw cardiotoxicity or nephrotoxicity. So I mean, it's the data from a collaborator, so I don't want to say something wrong, but they moved into phase 2. So they would not move into phase 2 if there were like severe toxicities. And our CAR T cell product, which we are using, which is in the clinical development for solid tumors, is mesothelin specific CAR T cells. And mesothelin is expressed in lungs. So what you see, or you might see in patients is lung toxicities, but we have a dose which is safe, a dose which we know you can apply to patients, which has an anti-tumor effect, but does not cause on target of tumor toxicities. So basically combining these 2 products, I think, based on the phase 1 clinical trials, is very promising, because we know that the STING agonist is safe, and we have a CAR T cell dose of these meso-specific CAR T cells, which we know does not cause lung toxicities. So yeah. Then another question from Takashi Nishina. I'm wondering how STING agonists enhance the trafficking of immune cells. Have you checked whether chemokine levels changed? Okay, so that is a very great question. So I think there will be changes of the chemokine receptors, but I did not check for that. This is something which we want to do now. So we want to look deeper into this. So I believe that probably if you look into tumors, after injection of the STING agonist, I expect that there are also changes in the chemokine levels, because the STING agonist obviously induces a very strong infiltration of different immune cells and also activation of different immune cells. And when you activate cells in a tumor microenvironment, you do not only get changes of the cytokines, which we saw, and I also presented today, I also expect to see changes in the chemokines, but I have not checked that yet. So hopefully soon I will be able to answer that question. Then let's see. What is the direct function of IL-18 to CAR T cells, from Phoenix Jiang? So IL-18, the effect of IL-18 on T cells, or specifically in CAR T cells, is something which the June laboratory, my mentor, works on that already like for like 10 or 15 years. So we know we have preclinical data on mice, on immunocompetent mice, as well as in immunosuppressed, like NSG mice, that the IL-18, the cytokine, significantly improves CAR T cell expansion. So the CAR T cells, when they get exposed to IL-18, they proliferate, they get activated, and they expand very well, and it also enhances their function. So we showed that in animals. There is right now a phase 1 clinical trial testing IL-18 secreting CAR T cells in lymphoma patients. And these patients, they received the commercially available CAR T cells, and they failed off the CAR T cells, the commercially available CAR T cells. And then they got a reinfusion of the same CAR T cells, basically, but with the IL-18 secreting, like they also secrete IL-18 upon activation. And we can see that those CAR T cells really perform very well. And the infusion rate, like the dose is very low. You see a very strong anti-tumor effect. I think they treated about like 20 patients now. It was also presented in the ASCO last week. So there should be also an abstract somewhere online if you're interested in that. But like IL-18 really seems to enhance the CAR T cells a lot. Let me see for more questions. So, did any of the mice experience complete remission in addition to their increased survival rates? So I think, okay, the time is like pretty early. So I think that was my first project, the surgery one, I believe. So in the surgery one, when we applied the CAR T cells intraoperatively following surgery, we got complete remission in all of the mice, basically. So in both NSG models, the xenograft models, as well as in the immunocompetent like 6 model. So pretty much in like 90, like almost all, I would say, like about 100 or 95%. Maybe one animal died, but not of the tumor regrowth. Like I think one died because it was spontaneous death. So sometimes you just find the mouse dead in the cage, although the tumor is cleared. Yeah, but it was basically 100%. Let me see. How to improve the infiltration of CAR T cells into solid tumors? Okay, so that's a very big field. So what clinical trials have shown, which works very well, especially in glioblastoma, in brain cancer, malignant brain cancers. There are like recent studies which were published in the few months, I think, is that the local infusion of the CAR T cells is very effective. So when you infuse the CAR T cells intravenously, they first need to traffic there. So they showed, actually, there are trials which tested glioblastoma with IV-infused CAR T cells. So the CAR T cells also get there, but when you apply the CAR T cells locally into the basically intraventricularly or intracranially, like the local infusion of those CAR T cells in glioblastoma patients, they really saw that the infiltration of the CAR T cells into the tumors is significantly higher compared to the clinical trials where they infused IV. So local infusion of CAR T cells is very promising, and it's also easier to do. But of course, there are also other engineering methods or strategies using synthetic biology. For example, there are different groups. There's also a clinical trial testing a CAR T cell. So the T cell was not only transduced with the CAR, but the T cell also is expressing a chemokine receptor, which basically directs the CAR T cells towards the tumors. So the chemokines are expressed from the tumor cells. There are like certain chemokines which is known to be expressed from lymphoma cells or from solid tumors. And they basically engineer the T cells to express the CAR, but also the chemokine receptor to make them go traffic better into those tumors. So I believe there is one trial which is currently also running, I think in China or in the US. I need to double check that. So let's see.
There's a question here. Ugi, did IMSA 101 induce intratumoral T cell death in your animal models?
Yeah, so that is a very good question, because STING agonists are known to cause T cell death. So there is published literature. So we looked into that. So we treated the mice also with the STING agonist. I took out the tumors, prepared the tumor single cell suspension, and looked also for the amount of dead T cells in those tumors. And we did not see differences in my animal models. So that was also because we know that the STING agonist might be toxic to T cells. We also basically first treated with the STING and then did the infusion. There is the second STING agonist dose was at day 3 after infusion, but we know that at day 3, the CAR T cells, they have not really trafficked yet into the tumors. So the peak expansion in the tumors is a week after infusion, like 7 to 10 days. So at 3 days, we do not expect a lot of CAR T cells. So that's why it's like very important, the experimental design. Also, when you want to translate the combination treatment into clinical trials, I think this is very important to consider, that you try to first give the STING agonist infusions intratumorally and then inject the CAR T cells IV to really make sure that there are not already a lot of CAR T cells in the tumors, which might also get killed by the STING injection, basically. Yeah, it's a very good question.
Great.
Yeah, Liam, please feel free. So if I forget any questions, I don't want to.
Yeah, sure. So, yeah, another good one here. Does the IMSA 101 affect any immune cells other than the T cells in the tumor?
So I looked for the T cells specifically. We looked for the CAR T cells, which I showed in the presentation, and also for macrophages. They get activated. I did not look for other immune cells, but I would expect that you also see differences in the other immune cells because the STING agonist is being injected intratumorally, and since you inject the STING agonist IT, you will just like basically activate everything what is in the tumors. In the flow cytometry data, we saw an increased trafficking of different immune cells. We can see significant changes in the cytokine levels. There are different immune cells who are producing cytokines. So I expect that those immune cells are also activated. NK cells, for example, they like IL-18, they like IL-15, and we also saw in the tumors a higher level of IL-18 in those tumor models, for example, and IL-18 is also needed to activate NK cells. So I also expect that the NK cells are activated, and also other immune cells, but we did not specifically check that. So I would need to confirm it also with data, but I would hypothesize that there is also activation of NK cells, macrophages, and other immune cells in the tumors.
Awesome. And I think in the interest of time, just that we have maybe a few minutes before the next session, we'll make this next one the last one, Ugi. But can you talk about any key factors or potential challenges moving forward in translating this treatment from these preclinical models to clinical trials for solid tumor treatment?
The key factors you said? I'm sorry.
Yeah.
Okay.
And key factors and potential challenges, maybe.
Yeah. So basically, I think we talked about that already a little bit. So the good thing is about these mesothelin-specific CAR T cells, which I used, it is already in phase 1. It is there. We know a dose which is safe for the CAR T cell product, and the STING agonist intratumorally injected is also already safe. They moved into phase 2 trials. So because both products as a monotherapy is safe, that will give evidence, or that will support basically the translation of a combination treatment. So if you knew that like one of the two treatments can cause severe toxicity, that will make it more difficult. But luckily, we know that like both products, there is a dose where both products alone are safe. And then you will still need to, when you combine treatments, I think the sequence of the treatment. So which treatment will you give first and which one will you give second? So these are things which you really need to think about. So the STING agonist might be toxic to T cells or to infused CAR T cells. So I think it would make sense to, I mean, that's how it will probably be the design, but the clinical trial has not started yet. So it is planned, but it hasn't started yet. So I would inject first the STING agonist in order to change the tumor microenvironment, because it obviously really does something to the tumors. It is able to change the tumor microenvironment and then infuse the CAR T cells. But you will really need to, like, that's a challenge. You will really need to think about that and like what sequence of the treatment and how many doses will be basically the best. Yeah,
definitely. All right. Yeah, really great answer. So Ugi, thanks so much for all your insights today and your presentation, as well as the Q&A. It's really been a pleasure having you with us.
Yeah, if anyone else has questions, please feel free to reach out to me on LinkedIn or Twitter, or email me. I'm always happy to discuss science.
Awesome. And so, yeah, thanks so much, everybody, for listening, and we'll see you again shortly at the next event.